this video we will discuss about this ALDH4A1 controls pyruvate import in the mitochondria. So this is the hypothesis that will be tested in this results that are presented in this figure. So first what I did is they checked this imported pyruvate in the conditions where ALDH4A1 is knocked down, and they can see that in the ALDH4A1 knockdown conditions they see a low import of pyruvate inside the mitochondria, and they checked it for different cancer cell lines such as HEP3B, HUH7, HEPG2, and in all the three cases they can see that there is a decrease in the imported pyruvate in the mitochondria when the ALDH4A1 is knocked down. And they also checked it in this NIH3 T3 cells, and here the Western blot shows that ALDH4A1 is knocked down and it is again introduced using the wild type ALDH4A1, and they can see that when the ALDH4A1 is knocked down they see a decrease in the pyruvate uptake in the mitochondria, and when they reintroduced this wild type ALDH4A1 they again see a regaining of the functions of this pyruvate intake. And so thereafter they wanted to check whether it has any role on MPC1 and 2, that is mitochondrial pyruvate carriers 1 and 2. And what it did is they for that they used the imported this 14C pyruvate, and this 14C pyruvate, and they checked at different conditions. So when they knocked down this ALDH4A1, they are able to see this imported pyruvates has been increased. The pyruvates imported into the mitochondria has been increased. That can be seen here compared to the controls. And when they inhibit this MPC, they are able to see a decrease in this mitochondria, indicating that MPC has a role towards this pyruvate that are imported into the mitochondria. And when they inhibit this MPC using the inhibitors, that is UK5099, they also see, in this case also they see a decrease in the imported 14C pyruvates. And in the case where they knock down only this MPC1, they can see the decrease in the imported 14C pyruvate. So 14C pyruvate decrease when the ALDH4A1 is not there, and also this MPC is been inhibited. And thereafter they check it the impact of this 4A1 when they reinstate this 4A1. So in that case, when it is knocked down, we see a decrease in the imported 14C pyruvate, and when they knock down the MPC1, they also see a decrease in compared to the control. But when they reinstate this wild type ALDH4A1, they are able to see a regaining of this 14C pyruvate functions. So this indicates that this ALDH4A1 has a role towards this mitochondrial import, and it is dependent on SHMPC1. And thereafter they want to find out the kinetics. They did a time dependent measurement of this pyruvate using Mendel's equations. I'm not able to recall, it is MM equations, and from that they calculate the velocity of this pyruvate import in the mitochondria. And they can see that when for the control they see a high velocity, that is 1.96, whereas for the case where ALDH4A1 is knocked down they see a decreased velocity, that is 1.12. and when they check this half time period for this pyruvate uptake for different conditions, they are able to see that for the control the half time period is 5.1, but when this ALDH4A1 is knocked down the half time period increases to 9.1. but when they reinstore or insert this wild type ALDH4A1, they can see a decrease in the half time period to 4.6, and when they reintroduce this mutated ALDH4A1 they see a decrease from 9.1 to 5.16. So this indicates that this ALDH4A1 has a reversible property, and it is has a role towards this mitochondrial pyruvate import in the mitochondria. Along with that, it also has a role towards this MPC, that is mitochondrial pyruvate carrier or mitochondrial pyruvate complex formations. So this is all that has been come up from the results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.